When I first started working on glioblastoma in early 2000, 2001, that was at a time when there was an enormous amount of pessimism about what could actually be done for glioblastoma patients. It was thought that the prognosis for the disease had not changed in 30 years, that there weren't any newer exciting therapies, and that people even said to me, "You're crazy to begin thinking about working on such a devastating disease." And I remember thinking at the time that, in fact, if we could only understand something about the signal transduction networks that were going on, meaning how the cells actually talk to each other, and if we could only begin to use that information to help guide the targeted therapies that we were beginning to see were so effective in other cancers, that perhaps we could begin to make a difference for glioblastoma patients. And during this period of time, I think due to the efforts of a lot of different talented people in the field, the landscape and the hopefulness about what can be done for glioblastoma patients has changed dramatically. And for reasons that I'll mention in a few moments, I think Accelerate Brain Cancer Cure has been a very critical part of that. There have been a number of, I think, key scientific findings over the last few years that have taught us the lesson that if you can understand something about the molecular composition of glioblastoma patients' tumors, you can direct the therapy to them that's most likely to benefit them. We've learned that with something called MGMT promoter methylation and response to chemotherapy and temozolomide. Many people have probably heard about that already, but it's a DNA repair mechanism, and it says that if that's disabled in the cancer cells, which it often is, those patients will do better when treated with radiation and chemotherapy. And I think a finding like that is hopeful because it says molecular composition matters and we can figure out who's going to benefit. Our work on the epidermal growth factor receptor, which I've talked about, has suggested that in fact we can use these pills that are really relatively minimally toxic. I won't say non-toxic, but they're not anywhere near as toxic as traditional chemotherapy agents or as radiation, because in fact they are so exquisitely targeted for specific molecules that those drugs could be targeted to patients based on the molecular composition, and in doing so, individual patients could come up with a management plan that could allow them to have maximum efficacy with minimum toxicity. And I think examples are already there that such a thing is happening. But we're only at the very beginning of this process, and in fact, the knowledge base is exploding. Because of these sorts of stories and because of other exciting work in the field, glioblastoma has gone from being a disease that most scientists didn't think much about or care about to being one of the most important or thought about diseases in all of cancer. At most of the major scientific meetings now, people are beginning to think carefully and seriously about glioblastoma. In fact, the NIH has just put a significant amount of resources in trying to sequence or understand the mutations that occur in glioblastoma patients. What all of this means is that we are in an era now where there's going to be an absolute explosion of information about glioblastoma, which is likely to translate, I believe, into a wide variety of new therapeutic options for glioblastoma patients. And central to that process will be developing ways to figure out how to use therapies that are targeted against specifically altered genes in cancer towards individual patients. And I think we're in a period, therefore, of enormous hope. And Accelerate Brain Cancer Cure has played a very important role in this process. When I started working in the field in 2001, 2002, I had some ideas about where we wanted to go with this, but they were premature for an NIH grant. But Accelerate Brain Cancer Cure was so committed towards helping investigators develop programs that could turn science into something for patients in the short term. I was funded by them 3 or 4 years in a row, and that in fact formed the basis for our work on the epidermal growth factor receptor that was recognized by the NIH as one of the most significant accomplishments in personalized medicine. And so that Accelerate Brain Cancer Cure has played a critical role in doing that and in bringing together top-level scientists from academia, from industry, from government to begin to sit together in a room and talk about what can be done for glioblastoma patients. So even though the disease is indeed a very challenging disease, we're in a period, I think, of unprecedented promise and hope for glioblastoma patients. There are already molecularly based treatments that are out there in clinical trials, and that in fact patients can get on those clinical trials at many of the major academic centers, and even some in the community that are affiliated with these, and that's something that can be investigated thoroughly and should be investigated thoroughly. And in fact, new combination therapies with drugs that I think have the potential to have much more impact on patient care, or in the planning or the early clinical trial stages. So I think patients who are now diagnosed with glioblastoma will have a variety of options that would have been unthinkable even 2 years ago. And that variety of options will include a series of clinical trials, everything from antibody-based vaccines to targeted therapies to combination therapies. And I think what this is likely to do is to have certainly a lot of impact for individual patients, and to help us all begin to learn how we can treat patients with glioblastoma in a much better way and in a much less toxic and a much more effective sort of a way. So I think it's actually a very important and optimistic time. And I think for a patient who's newly diagnosed with glioblastoma, now is the time to begin to gather as much information as possible, to find out what are the menu of clinical trials that are available, to ask hard questions. What are the toxicities that have been associated with it? How well do patients do on this? Are there molecular inclusion criteria or other inclusion criteria that I need to be thinking about to participate in these studies? What about the tissue? What tissue requirements do I need? How would I get my tissue to find out if I can participate in such a trial? Are there tests, molecular tests, that I need to have in order to participate in such a trial? All of those would not even have been questions 2 years ago, but we're in a period now where there's going to be a variety of new therapeutic opportunities for patients at various stages, some in the more mature phases, some in the early stages, and some just getting ready to start. So I think it's a very important period in which to try to be hopeful and to gain as much information and to think seriously about one's options.